Okay, yeah, we can go ahead and get started. I am recording this meeting, and so if you'd rather not have your face in recording, you can turn your video off. For those who are tuning in for the first time, I just want to give a quick intro to the MarineOmics Working Group and who we are. So myself, I'm a postdoc at Auburn, and my co-organizer, Sam Bogan at UC Santa Barbara. We put together this working group in order to promote reproducible and robust genomic research in marine science. We're part of the Research Coordinated Network on Evolution and Changing Seas. Part of our work in the working group is putting together these panels where we're bringing together experts to talk about a whole different range of genomic methods and analyses in non-model species. We're also, based on some of the recommendations we get from these panels, developing a website that will include recommendations for how to robustly and in a reproducible manner do these analyses. And the goal of this website is to be dynamic. So we're making some content on there, but we're hoping the community can add to it and help it grow, since this field is very dynamic and changing often. So today we're talking about a few types of population genetic analyses that you may do with genomic data, as well as things to consider when you're filtering your genomic data, whether that came from RADseq or whole genome sequencing. And so since this topic could be a seminar series in and of itself, today we're mostly focusing on ways to leverage experiments and genomic data to detect adaptation, considerations when you're conducting outlier tests or scans to identify loci under selection, and also principles of filtering. And then if we have time, we'll try to touch on considerations when you're actually describing population structure, like with PCA or admixture models. And so for the first 40 minutes or so, myself and my co-moderator, Joanna Griffiths, will be asking questions based on topics that we already sent to the panelists. And then we will open up the discussion to everyone with questions from the chat. But we do encourage participants to add questions and comments in the chat as they come up, and then we'll try to incorporate them in the current discussion, or if we don't get to them, we'll address them at the end. And so now I'm going to turn it over to another member of the working group, Sara Wuitchik, who's going to introduce our awesome panelists for today.
Thanks so much, Katherine. First and foremost, for our panelists, thank you so much for being here today. We really appreciate your insight and your expertise on these topics as we try to understand what some of the best guidelines and practices are in our fields. So today we have joining us Morgan Kelly, an assistant professor in biological sciences from Louisiana State University. Morgan's research interests include the ecology, evolution, and conservation of marine invertebrates, the genetics of thermal tolerance, as well as evolution of mating systems. We also are joined by Katie Lotterhos, an assistant professor in marine and environmental sciences at Northeastern University. Katie's research broadly addresses tons of different aspects of eco-evolutionary genomics using molecular ecology and population genetics. We're also joined by Zach Gompert, an associate professor in evolutionary biology at Utah State University. Zach's research program covers things like genetics of adaptation, fluctuating selection, and hybridization and speciation. And last but certainly not least, we have Jeff Ross-Ibarra, a professor in evolution and ecology at University of California, Davis, whose program encompasses evolutionary genomics, integrated genetics and genomics, and population and plant biology. So thank you again for being here today, and I will turn this over to our panel co-mediators, Katherine and Joanna.
Awesome. Okay, so feel free to either just unmute yourself and answer the question, or you can also use the hand raise question, the hand raise to ask or answer the question. Okay, so jumping right in, there are lots of different ways to answer questions related to population structure and selection and adaptation, such as combining different experimental methods from collecting multiple populations across a broad geographic ranges to in the lab manipulations of populations through different hybridizations and artificial selection experiments. And these can be combined with different sequencing approaches, such as pool sequencing, whole genome sequencing, so on. So how can we better leverage experimental designs to answer population genetic questions and identifying SNPs under selection? For just an example, what is one thing that maybe you wish you had done differently in the experimental design phase, but you had to mitigate in downstream analyses?
I guess I'll start us off. So a couple of things I would say. First, that I think the point you made at the beginning is one of the most critical ones, that I think combining multiple approaches is really kind of key. I think it's, if based on any one thing, whether it's spatial genetic variation, a time series, an experiment, any one thing, there's going to be severe limitations in your ability to really understand how natural selection is impacting the genome. But by combining those things, you have much more potential, right? So spatial analysis is nice because you've accumulated differentiation over time, but many processes are at play, so it's hard to really parse the relative contribution of different processes. A time series, you have a simpler sort of demographic scenario where you just get one population over time, but again, multiple processes going on and you're integrating over a smaller amount of time. Lab experiments, you have much more control, but you're integrating usually over even less time, unless perhaps you're doing some sort of many generation selection experiment in a system where that works. But you're able to isolate certain things, but you're also missing some of what's happening in nature, right? There might be selection pressures that play out in nature that don't play out in a given lab experiment. So that it's really sort of, I think, kind of a triangulation approach of having as many independent means of assessing selection as you have and trying to sort of bring those together. Along those lines, I think unless you're interested in something that you think is a single gene major effect type of situation, right, where you're really going to be able to detect strong selection on an individual locus or a few loci, I think integrating all of that with trait mapping is kind of critical, or something else to bring it all together. I think one of the things that's maybe happened in a bad way during the genomics revolution is that we've lost some of the earlier thinking about what understanding selection means, right? That if just saying, I detected selection on 20 loci, I don't think it really tells you very much. I don't think you've learned a whole bunch, but rather by understanding how both the sort of genotype phenotype map, and then how phenotype and environment interact to produce selection, that whole sort of combination of things is really more what we're after. And then how aspects of that environment vary in space and time that can cause those selection processes to differ. So really trying to integrate all those things in a multi-faceted approach, I think, is key. The other thing I'll say briefly, and then I'll turn it over for at least to somebody else, is that I don't think there's in any given case you're going to be limited obviously by total funds, right? So it's always a question of how do you allocate the resources you have across different strategies for experiments and sampling. And you can think about sampling the genome, right? You can think about sampling individuals per population, individuals per replicate, whatever it is you're thinking about, generations in time, additional populations. There's all these different dimensions you can play with, and depending on the specific thing you're after, different approaches are going to be differently important, right? Like you have to ask yourself, do I really need whole genomes here or not? Are they feasible? If you're doing trait mapping, probably the answer is no, they're just not feasible, whether you want them or not, because you want 10,000 individuals, and you probably can't afford to do 10,000 genomes unless you're working in a system where that makes sense. In other cases, pooled whole genome sequences just to get low frequencies makes great sense, and you can do that in a more cost-effective way. So it depends on the details of the question as you think about sort of all those different axes and how to maximize the outcome given what you want to do. And I think I wouldn't say there are times I've made what I thought of as big mistakes in that, but it's always something I think about up front. And so it's like I always want more. So yes, my mistake is that I didn't get more money, basically, to be able to do more of all those things. And it's just thinking about how you can best allocate those things and recognizing that there's limitations with any allocation scheme. Well, now I'll shut up for a second.
Yeah, go ahead.
Yeah, so I echo the importance of triangulating different approaches, and I like the what Zach mentioned, that I think a lot of people have sort of figured I'll just do a genome scan and find 10 loci under selection and are forgetting polygenic traits and quantitative traits. So I like that argument about triangulation. I think the mistake that I've made most often is not replicating things enough. Whatever the replicates are you care about, whether it's populations across space or individuals in some population that you really care about, or loci across the genome. Most often, it's number of, in the experiments that we do, it's most often it's total number of individuals or populations. And so I think there's sometimes a tendency to say, I want really awesome genotype data, so I'm going to sequence these to 10 or 20x coverage and do fewer individuals because of a limited budget. And I think more often you're better off doing 5 times as many individuals to 1 or 2x coverage for sequencing. I have gone in the other extreme, and we have a project that we've been working on now with individuals that are 0.5x coverage. And surprisingly, it is doable, and you can work with those data, but it's also much harder. So you can sort of go too far in the other direction. But I think thinking about what it is you're interested in and then replicating that, so for some projects where we were interested in quantitative traits, instead of having lots of progeny per mom, having more families, because the families really are the sort of independent replicates in that experiment. So thinking about your experimental design and what you want to replicate and getting lots of replicates, I think, is important.
Just one little brief comment to add to what Jeff just said about the coverage. I think I totally agree. And I think one of the key considerations there is thinking about whether your analysis is the level of genotype or allele frequency, right? Because if it's at the level of allele frequency in particular, you can go super low on coverage and you're just getting more information from more individuals. It's only when you're thinking about individuals that some minimum becomes reasonable. And even then, like you said, I think 1 or 2x is often going to do more for you than going to 10, 20x. There's just such diminishing returns as you increase coverage. As long as you're capable of modeling and propagating the uncertainty in genotype or allele frequency into downstream analyses, it's almost always better to drop coverage.
I'll mainly just echo more biological replicates, lower coverage. One mistake I think we made early on was doing reduced representation sequencing that didn't actually get us more biological replicates. It just got us more gaps between our loci. So it didn't actually get us more. So making sure that you're actually maximizing both things as much as possible. And then I guess I would add, very much agree with the multiple approaches. I would add, for some studies, gene expression data to that list of multiple approaches. There have been times for us in working with an organism that has a well-assembled genome, but the gene annotations with oysters are not great. And so oftentimes having gene expression data to sort of back in response to particular environmental challenges is another way to sort of back up that functional interpretation of loci that appear to be under selection.
I don't really have much to add. I agree with everything that's been said. And I think it really depends on the question that you want to ask. And you need to, I know my lab, and I think sometimes my students get a little frustrated because I'm like spending 6 months trying to talk through the whole experimental design with them before I let them do anything. So just be prepared to spend a lot of time like thinking through all the steps of the process all the way through, like, how am I going to analyze this data before you even start?
Awesome. Yeah, all really great advice that we all appreciate. Diving a little bit into some more detail. So pool sequencing in particular is becoming a common method for estimating allele frequency differences among different groups or treatments, especially in the marine field where researchers are working with hundreds of thousands of individual larvae. So what are some of the main differences or things researchers need to be aware of when they're analyzing their data using pool-seq methods?
I mean, really, the biggest thing in my mind is that so we got two things, right? So the biggest thing in terms of the analysis is that you're mostly losing genotype information, right? So you have to ask yourself, is that important or not? In some questions, it doesn't matter, right? That wasn't what you were going to do anything. You're just interested in the allele frequencies, and if so, then great. If you need genotypes, obviously it's not the way to go. Now, the other thing I would say is, you have to play so if you're kind of interested in genotypes, then it comes down to essentially the question of how much money are you saving on the reduced library preps, because you can always, right? So that's the amount of sequencing really doesn't change, or it shouldn't change. There's no reason to think that should change. It's really the library prep where it's making a difference. And are you taking the time to prep individuals, or are you just pulling them first? And then so the cost of library prep versus the cost of losing genotype information is the big thing. And then making sure that the models you're using account for the sampling, the distinct sampling design of pool-seq versus other approaches, especially if you're worried at all about unevenness in the amount of or quality or quantity of DNA coming from different individuals, which can cause more of an issue.
I'll add as well, specifically to your point, Joanna, like working with marine larvae, you can't prep individual libraries. It's just not worth your time. It's like not enough DNA. You have to have those pooled samples. So in the case where you're stuck with this, like I have to do pooling, what you want to do is design your experiment to maximize your confidence in what that allele frequency is in that pool. And so to do that, you need to think about how many individuals are in that pool and how deep does my coverage need to be to get that accurate allele frequency. Like if you only have 10 individuals and you have like 200x deep sequencing, you're not going to get a good estimate of that allele frequency because you don't have a lot of individuals. So you want to put as many individuals as you can into that pool. But there's a diminishing returns with respect to coverage, and maybe Zach has a better answer to this, but like, after you get maybe past 100 or 200x coverage, you're not going to get a much more accurate estimate of that allele frequency. You don't need to go super deep, but you need to think about, how many individuals are there and how deep do I need to go? And that needs to play into your planning and your budget.
Yeah, I totally agree. And I think that the ceiling is going to depend on number of individuals, right? As you have fewer individuals, you hit that ceiling even faster. 100x coverage with 10 individuals really isn't going to do a whole bunch. Now, if you have 1,000 individuals at 100, going up a little bit more will get you a little more precision, but there's still going to be diminishing returns. And unless you really need allele frequency estimates that are down to like, the third decimal point, you probably don't need much more than 100, 200x period.
We've also used pool-seq on larvae for that exact reason that you can't do individual library preps on larvae. I was wondering what the other folks on the panel think about with pool-seq. So you both have lost the ability to call the heterozygotes and homozygotes, but you've also lost that extended haplotype, right, that you would get from individual genotyping. How often is that something that seems like an important thing that you're missing?
We almost never work with pool-seq data, and we're fortunate that we don't have super tiny larvae. We are collaborating with folks that are doing sequencing on individual pollen grains. So there are approaches, if you have the funds to do cell sorting and sequencing at that level. The main issues that I think we have with pool-seq, first is the reusability of the data. There are a lot of times I've wanted to go back and use data from a study, and pool-seq data has pretty limited uses. It's great if you want allele frequencies, but as you say, if you want extended haplotypes or learn something about LB or genotypes or inbreeding or anything else, the data is fairly limited. And the other thing that I think is important to keep in mind is the different levels of sampling that you have in pool-seq data, because you're not just sampling alleles from individuals, but you're sampling individuals. And so different amounts of DNA coming from the different individuals go into the pool can lead to very different coverage of those individuals in your pool. And I've seen some pool-seq results where you get really strange results because only a few of the individuals contributed most of the DNA because the DNA extractions were somewhat wonky. And even when we do carefully per individual DNA extractions and titrate the DNA to be exactly the same, we get 2 or 3-fold variation in coverage sometimes. So I think that's something to keep in mind as well. There are approaches, sort of statistical approaches for doing things like inferring haplotypes from pool-seq data. If you have a set of known haplotypes from some other sort of analysis. But I think those depend on how well your known haplotypes are representative of what's in your pool.
Can I just ask for clarification, like if you sequence the parents, for example, of the larvae that you're sequencing, is that like haplotype information you can leverage?
Yeah, so if you have phase data from the parents, for example, then the pools of the progeny should allow you to infer frequencies of the haplotypes in that pool.
Yeah, cool. Cool. Well, yeah, thank you. And so moving on a little bit to filtering your data. And so 2 weeks ago, we had like a whole genome-seq panel where we talked about with low and high coverage, you end up with genotype likelihoods for SNPs or hard-called SNPs. So once you get that VCF file or genotype likelihood file, what principles do you guys use when deciding how to filter your data, irrespective of downstream analyses? And then we'll get into how you decide to filter that data depending on what you want to do with it.
So I guess the first thing, I mean, so it depends on the kind of data, right? So for RAD-seq, GBS, anything of that nature, I think the first things I think about are having the sequence data from a SNP present in the vast majority of individuals. And I think that's really important to avoid the issue of restriction site polymorphism that can really mess things up. That you don't have to worry about so much with whole genome data, right? With whole genome data, if it's low coverage and you're missing a bunch of data, if you're doing like low frequencies, it's kind of all okay. With GBS or RAD-seq, there's a good potential for linkage disequilibrium between a SNP in the cut site and a SNP in the sequence. So the missing data is unlikely to be random. So you really want to, if it's truly missing, right, like no data from individuals, you want to get rid of stuff. My standards, like if it's not present in at least 80–85% of individuals, it's out. And I don't go much lower than that ever. And then aside from that, I usually set some minimum average coverage, 1 or 2x, usually not something much higher than that. And then depending on the kind of data, there's various calculations of bias that you can use that are often present in the VCF files depending on how you process them, and some of them make good sense in certain cases, like things for bias in whether the reference versus alternative allele shows up in different positions in the read more often than you'd expect by chance, or different qualities associated with those. And I think all of that to me is kind of getting at whether you're potentially dealing with things that are paralogs, right? Where you have things that are not really the same assembled into the same place, and those are good to filter for. You have to be careful because in some cases with some kind of sequencing designs, there might be like for example, if you're doing GBS or RAD-seq, especially if you're doing a double digest, the SNP is always going to be in the exact same place in the read, so it's kind of, well, that's not going to tell you anything, right? So you have to think about the details of your sequencing design, and in some cases, what might be a cause of concern with one sequencing approach might be exactly what you'd expect with another. So you have to be a little cautious of that. And then I also filter for excessively high coverage. I usually don't apply any sort of minor allele frequency filters, at least not right away. Maybe for certain downstream analyses, but not as a general rule.
Also, I will hop in just because in our RAD-seq panel that we had 4 weeks ago, we actually talked a lot about missing data and had a little takeaway of embrace the missingness. So yeah, I think it is sort of a like active area of debate maybe of what you're doing to the data when you filter for too much missingness or not enough missingness. But yeah, no, it's good to get another sort of contrasting perspective.
Define it too, right? So when I'm saying missing, I mean no data for an individual. I think low coverage, like not being able to call a genotype, that's not missing. But when you have individuals that are, when you have a locus where you only get data for a subset of individuals, that's what makes me more nervous.
Okay, yeah. No, that's a great point. Yeah. I think we were talking about if you had loci found in lots of individuals, but there's lots of individuals missing that locus. Yeah, no, that's great. That's a great point. There's different kinds of missingness. All right, yeah. So other thoughts about filtering. Does anyone have, I guess, a different strategy than Zach?
So the one place that I actually might disagree is that we find a huge impact of filtering on missing data, even in whole genome sequence data. And some of that is because, as you guys have said, the different kinds of missingness. So, something can be missing because of stochastic sampling, but often, at least in the species we work on, it's missing because of biological presence-absence variation. And that can also impact your ability to map reads, et cetera. So we usually filter for something like 80 or 90% of individuals having data at a site. Other things that we found really important is mapping quality because of the repetitiveness of many genomes. The number of base pairs in a window. So when we actually come down to start doing population genetic statistics, if you're doing 10 KB, 1 KB, 50 KB, whatever your windows are, we usually dump windows that don't have data for less than some percent of the window, because we find that those give really inaccurate estimates of pi or whatever the statistic is you care about. And then in addition to the things previously mentioned, we have for really diverse species, you can often see a big impact of reference bias where alleles that are different from the reference won't map. And one way to look for that is allelic balance, which is a statistic often given in some of in BCF formats, which is in heterozygotes, you expect that 50% of the alleles are A and 50% are B. But if you see that across all of the heterozygotes in your sample, 80% of the reads come from A and 20% come from B, that suggests that the B haplotype, for whatever reason, isn't mapping as well. And that SNP, you're probably not getting good information from that SNP. And so we often will filter on allelic balance as well.
Yeah, I'll just echo that idea that you need to know your reference and how good it is really well. So it was a couple of examples. This wasn't something my lab did, but I know in one case, someone was mapping to a reference genome that was quite distant from the species they were studying. And so the species they were studying had SNPs, but the reference genome actually had a third nucleotide. And so it looked like they were triallelic sites, which are things that people quite often will filter out. But it was actually a SNP in the data. It was just that it was called triallelic because the reference had a third nucleotide. So that kind of thing is something to be aware of and think about.
One short thing I'll add to that we found in cases where I've had similar issues where you have one, where you have multiple species you're dealing with and one's closer reference to the other ones are not, you can do an initial alignment to the reference for, so take species X that's not close to your reference, align to it, and you can basically create a new consensus reference that essentially replaces the bases in the original reference with the most common base in your that align to it, and then redo your mapping to that. So that you essentially take a reference, you're not changing the order of anything, so it still has the structure of the original reference, but you're turning it into as close as you can get to what the reference would be for the species you're using, then realign to that, and it helps with that. It doesn't fix it. You're still not going to get as much aligning as you would if you had the right genome, but usually you can get you get 2 to 3 times maybe as many reads aligning as you would otherwise by doing that correction first.
Yeah, I think that came up in the 2 weeks ago too, didn't it, Catherine? That we were talking about that. Yeah.
Yeah.
The other thing I was going to say about references is like when we were working with the Eastern oyster, we found that there was a lot of what we qualify as misassemblies. So when you assemble a highly heterozygous genome, you want, a diploid genome, you want both of those, diplotypes to be in the same place on your map. But what was happening, it was so heterozygous that we had these haplotigs. So it was really the same place in the genetic map, but they were mapping to different places on our reference. And we saw that reads were going down and then up in some places and down in some places because they were split. Half of the reads went to one haplotype and the other half went to the other haplotype. And so, we've developed a much better reference from that. But just looking at coverage and patterns of coverage across your genome too, it helps make decisions about how to filter.
I'm muted. Great. And so now digging in a little bit more into specific analyses and how you might filter. For example, if you wanted to estimate nucleotide diversity statistics, what can throw off those estimates if you don't filter your data correctly?
This is maybe a bit tangential to filtering, but the soapbox I end up usually getting on when reviewing papers is for anything where you care about per base pair values like nucleotide diversity. A really common problem that I see is that people ignore invariant sites. So you get a VCF, you call all your SNPs, you put it in VCF tools and you estimate pi. Much software, VCF tools and many other software packages assume that if you're doing some window-based statistic, I have my 10 KB window, that you have sequenced every single base pair in that window. And so it counts up the number of SNPs, estimates diversity, and divides by 10,000. But if you have windows in the genome that vary in how much sequence that you've been able to actually assay because of, say, transposal elements or simple repeats or something like that, then the denominator for per base pair statistics varies across windows. And I've seen many data sets where the correlation between the pi values that they estimated and the true pi values are essentially zero, because all of that variation is explained by the number of base pairs that you could actually assay in that window. And this is something you need to think about before you call SNPs, because you need to be able to identify sites in the genome which are invariant in your sample, so that they're monomorphic in your sample, but are high enough quality that you could have identified a SNP if there was one. And there are a number of approaches for doing that, but missing or not thinking about invariant sites is actually, I think, a common error.
I completely agree. I think that is the error basically. I mean, that's the thing that's going to kill you with nucleotide diversity and like Jeff said, it's not just did you get it at all or not, but did you get enough sequence coverage at a site to be confident it's invariant? And it makes me nervous and skeptical in most cases of pi estimates. Like it's anything where you have to divide by the number of loci is much trickier with next gen data than it was back with Sanger data.
Pi confused me a lot with my first RADseq data set. I was like, I don't know how to estimate this with anything that's available. So I just didn't. Great. And so now thinking a little bit about population structure methods, such as PCA or admixture models. Katie, I think you maybe had a quiz or something for the panel.
I have two challenges for the panel. So see if you guys can figure out what's messing up these. Okay, so I'm going to share my screen. Okay, can you guys see that?
Yep.
Okay. So this is a data set that was used in a workshop that I attended one time. So it's cichlid fish from 3 locations in Lake Malawi. And with replicate runs of essentially what's the equivalent of structure, you get these 2 configurations. So what do you guys think? Why do you think we get these 2 configurations from structure? What's messing it up?
Are they different?
They're the same. It's the same data set.
No, but the 2 they don't look different to me. Like,
well,
problem. I don't think it's just the colors are wrong. Is that it? I mean,
no, there's one, like the second sample is something different.
It's mild, right? Okay, there are. It's mostly.
So it's instead of 3 clusters, red, blue, and green, we get 2 clusters of red, and then a blue, and a third cluster is here.
Yeah. But yeah, although, yeah. Okay. Never mind. Sorry. Yes.
Yeah, I think they're different.
Yes, no, they are different. I was looking at the left part, and it was like, oh, it's just the colors. But no, you're right. Then it doesn't get split or not. I mean, so the short answer is they're different because you're not getting convergence. I mean, that's the bottom line. You're not.
But there's something about the data that's causing the problem.
Yeah, I mean, you're not converged, or well, sort of, right? You're not converged. You haven't actually converged to the posterior distribution and sampled it well. In other words, those are not the actual posterior distributions, right? You haven't adequately sampled the posterior. And presumably it's because there are two modes that are not that different, and you're getting stuck on one of the two modes each time.
But there's something we could filter the data for that would prevent this from happening.
So not, I mean, maybe, but it may just be reality too, right? It might be that there's two solutions that are not that different in terms of the sort of likelihoods, right? So this is both the strength and a problem of structure so anytime you run structure or something like it, there's actually many modes. There's inherently many modes because there's a label switching issue that, you could with K equals 2, it could be these guys are mode 1, it's like one solution is this is species 1, this is species 2, the other solution is flipping it. And if structure actually mixed really well, it wouldn't work at all because you would explore all those modes and it would all break and you'd always get a rainbow. Structure, it gets stuck usually on one mode, which is a good thing. And you're able to sample that and actually get a reasonable posterior. In this case, you've got additional modes and it's still getting stuck on one of the modes and not going to the other one. You could change some of the actual MCMC parameters. You have access to that in structure. You'd have to be careful. You don't want it to start mixing so well that you make the rainbow, right? You want it to be able to move between these two modes and not all the modes.
Yeah, Jeff.
I mean, I don't know is the short answer, but I'm going to guess it's something with the genotype data, either missing this, or we've seen similar results when we have a bunch of really bad quality SNPs, and that additional filtering for SNP quality has really cleaned up at least the ability to replicate structure plots.
Nope. Morgan, you want to take a guess? Okay. So these two individuals are siblings in the data. And so sometimes structure clusters these two individuals as their own cluster. And so relatedness, having varying degrees of relatedness is a source of non-independence that can screw up some structure-based estimates. Principal components I've also seen is really susceptible to this. If you have a group of siblings or a group of highly related individuals, they can really warp those principal components. So if we look at the principal components for this, these two individuals here are the two siblings. So if you remove one of those individuals, the problem goes away. You don't have that problem anymore.
Which again, so it goes back to, it really is that you have two modes then, right? So it's not error. It's that when you put those in there, that is another good solution and it's getting stuck on that solution sometimes.
Yep, exactly. But there's a simple solution that you don't even need to figure out the program. You can just remove one of a related pair and not confuse that anymore. But it depends on your question. If you're doing genome scans and your genome scans are dependent on having a very accurate estimate of structure, then depending on the method, that could screw it up. Or if you're doing like a genome-wide association study, you're including that pedigree structure usually in controlling for it in that association test. So it depends a lot on the method that you're using and whether or not you're accurately capturing those relationships.
I think it does depend too on your question, because you see the same thing with levels of inbreeding across populations. That if you have one highly inbred population, that will often come out as the first separation at K equals 2 or 3 or something like that.
Yeah.
We've also seen problems where structure or PCA will separate based on coverage of the data, which took us a long time to figure out, where the high coverage samples get separated into different clusters because you have high confidence in those SNPs.
A quick related question from the chat. What thresholds could you use to remove related individuals or clones?
That's a great question. I haven't explored that. Has anyone else here explored that? I
mean, we've played with this some, and I think, again, it depends on your question. If you care about if you care about relatedness and inbreeding, then that may change your answer. But we will often, in sampling from real populations, we're either careful to sample seeds from different mothers, so we can be assured that individuals are at least half sibs. And then we often will filter to remove half sibs or sibs, anything sort of at that level or above, figuring that sort of recent relatedness is not something that we care about. But sometimes you may care about that if you want to know about biparental inbreeding or other sorts of processes.
And just a quick comment from Paul Moran that removal of full sibs can certainly bias some PopGen metrics. But yeah, for the sake of time, Katie, do you want to move on to some of your other ones?
Yeah, so I just had 2 challenges. This is the second challenge. So in this challenge, for this data, I simulated this landscape with 2 mountaintops, and there was adaptation, and there was isolation by distance population structure. So this is what I expected. But when I put the data into the program, I saw this observed population structure. So what did I simulate in the genome that caused this to happen to the principal component? So this is not a SNP quality problem. These are simulated data.
How did you sample? Did you sample uniformly?
I sample uniformly. Mm-hmm.
Did you simulate?
I put all the data from the genome that I simulated into the program.
Is this a lack of recombination in your sims?
There is recombination variation in the simulation. Wes Larson got it. He's in the chat. He said inversion. Yeah, it was an inversion, but it was a neutral inversion. So it wasn't involved in adaptation at all. Even though the inversion, so I simulated a bunch of different stuff. There was a neutral inversion, a centromere in these simulations on 2 linkage groups. And the SNPs only took up like 5% of the data, of the total SNP data, but it still really drove the principal components if you used all of the data in the genome scan. And I've seen it really drive other ways of estimating and looking at structure as well. So, when you're thinking about doing some of these outlier tests, you really need to ask, are you really accurately estimating the structure, and is that being influenced by some degree of non-independence? So, we've gotten a lot better with methods looking at complex demography and controlling for that. But there's also a source of non-independence on whether loci are linked or not in the genome. And this is a source of non-independence that can really screw up principal components. So in this simulation study, when I thinned SNPs for LD first, used that thin set of SNPs to do neutral parameterization, which is estimate the nomography for those principal components based outlier tests, and then go back and do the genome scan on all of the SNPs, you got a much more accurate estimate. So I didn't say earlier, but because this method called PC Adapt works on principal components, if it thinks this is the structure, it was also saying the inversions and the centromere were the outliers, like the FST outliers. But with this best practice, where you filter for LD first, and then you estimate structure, and then you go back and do the genome scan, you get outliers in the locations where the markers that affect the phenotype that is under selection in the simulations are, which is where these orange arrows are. So that's another thing that, for people that have inversions in their data set, they need to be aware of, filtering in this manner as well before you conduct these tests.
Yeah, and as Jeff mentioned in the chat, the flip side is that you can use the signal structure to actually identify inversions, which is also pretty cool.
We've done that too.
Yeah, I think Joanna has some questions about LD and outlier test. It's a nice segue.
Yeah, the main goal in a lot of population genetic studies is to identify loci under selection, either in the wild or a laboratory environment. However, these tests are often plagued by really high linkage disequilibrium. So how can we best account for LD when identifying SNP outliers? And are there any suggested methods, either sequencing or downstream filtering, to disentangle outlier SNPs in LD and SNPs under selection?
So one thing I think you have to keep in mind is that, I mean, this is kind of what you're saying, but you know, selection, that you have selection acting directly on some subset of loci, but because of LD, you're going to have indirect selection as well. This was something that was recognized back in the 70s and 80s with old analysis of phenotypes. And there they would just use multiple regression to try to tease out what was directly versus indirectly selected. It doesn't work, of course, with genomic data because you don't have a sample size that's larger than the number of loci. So you can't use those standard approaches. And it's really a big deal in the sense that I think in any reasonable case, pretty much every locus in the genome is going to be indirectly affected by selection. It may be weak, it may be very, very weak, but LD between a causal variant and if you have more than a few causal variants, almost every locus have at least some small non-zero value of LD with it, just for stochastic reasons. And the same reason, it's similar to the reason that drift will happen in any finite population, right? It's a mathematical necessity. There'll be some small amount of LD. So everything basically is being affected by selection. So it's relatively easy to try to estimate the strength of total selection experienced by a given locus. There are methods for doing that. I think going from that to try to identify the actual causal variants, the actual things under direct selection, especially in cases where it's polygenic, is really, 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 really hard. I think you know, if it's a major effect locus, I think you have some chance to narrow it down to small, you know, to try to identify the actual causal variant and maybe eventually do some sort of genetic manipulation to say, okay, no, this is the thing, this is the directly selected thing. But for polygenic selection in particular, I'm not sure it's the right thing to try even. I think it's trying to disentangle being affected by selection because of LD from being causal. You have to really ask whether that's, I think, an important thing to do in a given situation. So I think it's really, really hard.
Morgan, did you unmute yourself to say something? Oh.
I think I was just accidentally unmuted. Agreed with all of those points, that asking whether you actually want the causal SNP or if it's possible is a really important thing to ask yourself.
Yeah, I think that's a great point. But I mean, as Katie showed in her simulations, you know, even if you just are trying to find initially regions of the genome that show outliers, there are some simple approaches just on downsampling SNPs or filtering based on distance or LD that you can use to help avoid sort of egregious false positives. But totally agree that for many things, you may have no hope of finding the causative SNP, or it may not actually be important to find the causative SNP.
Yeah, and so then, you know, another question we have is related to identifying outliers, is how can demographic and experimental structure mess up these outlier tests, and how can we account for it? And so I think Katie touched on this a little bit with like siblings, but is there other sort of demographic or weird sampling that can happen that can throw off these outlier tests?
One thing I've heard people be concerned a lot about is when structure coincides with the adaptive signal that you're trying to detect, or like when you correct for that structure, are you potentially overcorrecting and losing a signal? And I haven't seen a good, like, thorough treatment or answer to that question in the literature. I don't know, are you guys or other panelists or people on the call aware of anything?
So I think that certainly happens. I guess it comes down to what you're trying to do. So we see in plant populations, because timing of flowering is one of the things that reproductively separates populations. If you're interested in looking at selection on flowering time, that invariably is essentially collinear with population structure. And so correcting for population structure, you can't identify things that are selected for selection on flowering time. But you can get around that by doing, experiments and growing things in a common garden. There are some sort of permutation approaches you can try to think about, but generally speaking, actually doing some experiments and measuring phenotype is one way around that.
Yeah, I mean, I think related to that, right, the general idea, I think, if you're doing some sort of genoscan antler test, at best you should be thinking that you're finding candidates worthy of some degree of further investigation, but then some sort of experimental follow-up is the right answer for how to go beyond that.
Something else that I worry about with structure that I don't have a good answer to with marine invertebrates is that there is often also structure among cohorts of settling larvae from one year to the next. And when you sample full-size adults, you could easily be sampling different year classes of larvae that may have come from very different places. And you don't even necessarily know that when you're sampling full-sized adults. I don't have a good solution to it besides maybe being very careful to sample same-aged individuals or to know the age.
Yeah, and also like sweepstakes, reproductive success that can just with drift vary, what genotypes are in each cohort. Yeah, something I've also worried about.
So we have some questions in the chat, starting with Kyle. When filtering VCF files, how do you decide what cutoffs are appropriate for your minor allele frequency cutoffs? He's working with LCWG data of 9 oyster populations, 20 individuals for each pop, and 4x coverage.
I think it depends some on the question, but for a lot of questions, I think the answer is 0, right? As long as it's there, because it's relevant variation. I do think there's benefits, for example, of looking at repeating analyses with different subsets of loci, including rare variants, excluding them. But there's no reason a priori, I think, for most applications to throw out rare variants.
Yeah, I concur. I think we almost never use minor allele frequency cutoffs. I think the exception would be if you were worried about genotyping error, you might remove singletons, but removing singletons removes information about demography and genetic load and other things you might care about. And the other exception is when using genotype likelihoods, we'll often filter to remove allele frequencies that are impossibly small. So if you have a sample of 20 individuals, you shouldn't have an allele that's at a frequency of 1%.
I'm going to disagree I agree, but I'm going to disagree. Like with FST outlier tests, you have to filter for minor allele frequencies. We know that the minor allele, like really low, less than 1% allele frequencies, really rare ones will really mess up some of these tests. They don't follow the statistical properties or statistical assumptions that the tests are based on. So when you're doing some outlier tests, you definitely want to filter for minor allele frequency. I've seen it in simulations, so I know it's not just like something that happens in real data, but it's mostly really low, like less than 1%.
Yeah, I would say with RAD, this is getting back to the singleton genotyping error. It can clean up your population structure, your PCA a lot if you remove like less than 1% or less than even 5%. Just because then you're getting the SNPs with the strongest structure, strongest signal. That's what I've seen. I actually had a question from earlier. This is just about pooled seq, what are folks' thoughts on population, which is this really popular R program, or are there other preferred methods for getting those allele frequencies from pooled data? Yes, for like Morgan, I know you've done some pool-seq stuff.
Yeah, that's what we've used. I'd be curious to hear if anyone else has any other preferred methods, but yeah, that's what we've used.
Cool. No, I mean that's great. If that's what the standard is, that's good to know. Are there any other questions from the group that people want to throw in the chat? Nope.
Here,
Cool.
question from Gabe on here. He's asking, how do you correct for small sample size bias with a clustering to larger populations?
Gabe, can you tell us a little bit more about your application, specifically like
Says PCA and structure analysis.
Maybe I'm not quite sure what the question is. Is the concern that sample size is biasing the PCA result?
You can also go ahead and unmute yourself, Gabe, if you prefer to just talk about it.
Yeah.
Oh, yeah. Sorry, guys. Yeah, so having population stratification not being shown through the PCA and having it clustered to the larger population. Do you guys remove or subset the larger population, or do you remove certain SNPs?
I think if you have because right, so PCA, and the same is true of structure mostly, it's going to first find those sort of axes that explain most of the variations. If you have a small population, it's unless it really stands out in some way, it's unlikely to be one of the first few axes. So if you want to sort of get rid of that effect, then yeah, drop individuals and put them all in sort of equally, and then it should pop out sooner. Presumably right now it's like PC6 or something, right? If you kept going, it presumably comes down at some point. It's just not going to be the first few.
And those other PCs are still really informative for pulling out those groups. We have a question from Marie Strader. We're working with a species that potentially has cryptic multiple species within it, and also maybe clones. So what might you recommend for developing a population study in terms of methods and depth of coverage?
I'd be curious to hear people who work with the low coverage data on this question. Like if you're sampling a bunch of individuals and you don't know what group, a priori that they're going to belong to, would you still recommend low coverage for that scenario? Do you think it would still be good at identifying, cryptic species and clones?
Yeah, I mean, we faced this situation with some polyploid clonal grasses that we're working on, where we have no idea a priori what the ploidy is or what the structure is. And I'm still a fan of low coverage, lots of individuals. With 1x coverage genome sequence data, you can readily identify things that are very, very different from each other. And this gets back to the point about replication, is that if I think a priori that I'm going to get some clones in the data, I'd like to just toss those, because those aren't independent sort of genotypes. And if I only have 5 samples and I lose 3 of them, I'm in trouble. But if I sampled a bunch of individuals in the population, I'm in better shape if I have to dump half my samples because they're clones or something weird.
And I have one more question from our list actually is, how important is it to remove outlier loci before looking at population structure? And then relatedly, what's the utility of analyzing those loci sets separately? For example, you pull out 100 putatively adaptive loci, you see some population structure with that, you don't see anything with the neutral loci. What does that tell you?
I think this is a question that has been coming up in like marine fisheries literature a lot.
Yeah.
And I think Atlantic cod is a good example of this. So in different parts of the species range, there's these like cryptic ecotypes of cod. Sometimes they're spatially separate, sometimes they're in the same place, like differences in spawning time or differences in like migratory behavior. And population structure on genome-wide loci show no differences between these ecotypes. But when there's a lot of evidence that the basis for the differentiation is in inversions. And in that case, when you look at the inversion, you know, it pretty strongly separates the two ecotypes. So, you know, do we manage the population based on the genome-wide signal, which says it's one population, or do we manage the population based on, you know, the actual loci that are causing the adaptive change, which says these are two different populations? And my opinion is, yeah, we should manage it based on the adaptive loci. But I think that's still something that people debate.
Yeah, I mean, I think basically, sorry. So you know, it depends. It depends a lot, or to some extent, on what those things are. It could be those few things in the tail of the distribution are an inversion. Maybe there are a couple inversions. Maybe it's the sex chromosome, or maybe there are things spread all over the place that are important for adaptation. Maybe none of them are important for adaptation. You don't really know what they are unless you do a little bit more, right? Like saying, okay, yeah, it's in an inversion. In some cases, like Katie was just talking about, well, then you see, okay, that is the thing, maybe. And then I think, yeah, like that's important, and it makes sense in it from an applied context, managing based on the thing that's important, just in the sense that you wouldn't manage species based on the entire multidimensional phenotype. You manage them based on like the things that are different and that are important, right? In other cases, maybe they're not, you know, it's even if the inversion is not important, though, there could be still an inversion. That could still be your outlier loci. And you could get, oh yeah, if you manage based on that inversion, it's actually doing nothing because it's just some neutral inversion, right? You can still get the structure. So it depends on what you're trying to accomplish. It depends on what you're trying to do. There's not a single answer, I think, and it depends on the distribution. Like, are the outliers, like, is it part of the continuous distribution, or are they really these different things, and why?
So you think it's still worth reporting, at least, and still worth looking at them separately, if only so someone else can maybe figure out what exactly is going on with those loci?
I think it can be, although my personal opinion, my personal biased opinion, is that if all you're doing with the outliers is saying, hey, they're outliers and look at them, I think that's not a useful activity, period. Unless you're comparing them to something else, doing something more than, I think it's a waste of time. Just a basic outlier scan, I don't think tells you anything useful, other than the distributions have tails.
So you don't think that there can be like follow-up research on those outliers?
Sure, I guess if it's planned, if it's really gonna happen, right?
Oh, yeah.
Actually gonna have follow-up research, fine.
Yeah.
I mean, I think the thing you have to be careful about is the circularity of the question. Is like, some outlier tests, it's like, I'm saying this is one population and I'm saying this is the other population, and it's telling me what's different between those populations. And then I'm going to put what's different between those populations into a PCA, and then it's going to tell me that those populations are different, right? So
yeah.
You have to be aware of the potential for circularity when you're doing that exercise.
Yeah, I think that's my big concern with it, is it feels very circular, but it's also something that I've even done, and I've seen a lot of people do. And so, yeah, what is the real utility?
I think it'd be a fun exercise to try making essentially, you know, artificial populations, splitting them and finding the outliers, and see how often you could recover those populations, right? And to see if at least you're doing better than that with your real outliers.
Paul Moran has a question about how do you study epistasis in NGS data?
Paul can also unmute himself to maybe give a little bit more detail. Like, is he looking for like programs or specific analyses or even experimental design questions?
Like, so so study epistasis in what sense though, maybe? Like trying to, for example, like trying to find cases where trait variations explained by epistatic interactions between markers, or you're thinking about epistasis for fitness, or in what kind of context?
Paul, are you still on? Oh, yep, he's here. Paul, do you want to unmute yourself? Okay, well, we can, yeah, if he chimes in, we can move on. We have just a few more minutes. Are there any other last questions for our panelist? And if not, then thank you all so much. That was great. That was really helpful. This is recorded and will be up in about a week on the website. And then in 2 weeks, everyone, we will have a functional genomics panel seminar led by my co-organizer, Sam Bogan. And so make sure that you tune in for that. It will be focused particularly on RNA-seq. So yeah, a big round of applause for our panelists. Thank you guys so much.
Thanks, Katherine. That was fun.
Thanks,
Yeah,
Joanna. Thanks, Katherine.
Great.